Cell is the fundamental unit of life responsible for carrying out essential biological functions. Cells contain various organelles, each with a specific role in maintaining cellular processes and overall homeostasis. These organelles work in coordination to support metabolism, growth, communication, and genetic regulation. Mitochondria. Mitochondria are double-membraned organelles known as the powerhouses of the cell. They generate adenosine triphosphate through oxidative phosphorylation, providing energy for cellular processes. Mitochondria have their own DNA, allowing them to replicate independently of the nucleus. They play a key role in metabolism, apoptosis, and calcium signaling. Their number within a cell varies depending on the energy requirements of the specific cell type. Plasma membrane. The plasma membrane is a selectively permeable lipid bilayer composed mainly of phospholipids, proteins, and carbohydrates. It regulates the movement of substances in and out of the cell, maintaining homeostasis. The fluid mosaic model describes its structure, emphasizing the dynamic movement of embedded proteins. The membrane also facilitates cell signaling and communication through receptor proteins. Additionally, microtubule. Microtubules are part of the cytoskeleton, providing structural support and shape to the cell. They are composed of tubulin proteins and play a crucial role in intracellular transport, cell division, and maintaining organelle positioning. Microtubules form the mitotic spindle during cell division, ensuring proper chromosome segregation. They are also involved in the movement of cilia and flagella in certain cell types. Centrosome. The centrosome is an organelle that serves as the main microtubule organizing center in animal cells. It consists of two centrioles surrounded by a pericentriolar matrix. It plays a vital role in cell division by organizing the mitotic spindle and ensuring accurate chromosome segregation. The centrosome is also involved in cell polarity and intracellular trafficking. Defects in centrosome function can lead to chromosomal instability and diseases like cancer. Microfilament. Microfilaments, also known as actin filaments, are thin, thread-like structures that contribute to the cytoskeleton. They are composed of actin proteins and are essential for cell movement, shape maintenance, and intracellular transport. Microfilaments play a role in muscle contraction, cytokinesis, and endocytosis. Their dynamic nature allows them to rapidly reorganize in response to cellular needs. They also facilitate cell signaling. Lysosome. Lysos are membrane-bound organelles responsible for the degradation of cellular waste and foreign substances. They contain hydrolytic enzymes that break down macromolecules like proteins, lipids, and carbohydrates. Lysosomes play a key role in autophagy, the process of recycling cellular components. Their acidic internal environment is essential for enzymatic activity. Dysfunction in lysosomes is associated with lysosomal storage diseases such as Tay-Sachs and Gaucher disease. Smooth endoplasmic reticulum. It is a network of tubules and vesicles that lacks ribosomes on its surface. It is primarily involved in lipid synthesis, detoxification of harmful substances, and calcium ion storage. ACR plays a role in carbohydrate metabolism and steroid hormone production. In muscle cells, it functions as the sarcoplasmic reticulum, regulating calcium levels for contraction. Its structure and function vary depending on cell type and physiological demand. Cytoplasm. The cytoplasm is the gel-like substance that fills the interior of the cell, excluding the nucleus. It contains cytosol, organelles, and various biomolecules necessary for cellular function. Cytoplasmic streaming facilitates the distribution of nutrients and organelles within the cell. It serves as the site for numerous biochemical reactions, including glycolysis and protein synthesis. The cytoplasm also provides structural support and contributes to cell motility. Golgi apparatus. The Golgi apparatus is a series of stacked membrane-bound sacs responsible for modifying, sorting and packaging proteins and lipids. It receives proteins from the rough endoplasmic reticulum and processes them for transport. The Golgi apparatus plays a crucial role in glycosylation, a modification necessary for protein function. It produces lysosomes and secretory vesicles that transport molecules to their destination. Dysfunction in the Golgi apparatus can lead to protein trafficking disorders. Golgi vesicle. Golgi vesicles are small membrane-bound structures that transport materials within the Golgi apparatus and between other organelles. They facilitate the movement of proteins and lipids through the Golgi stacks. Golgi vesicles play a role in the secretion of enzymes, hormones, and neurotransmitters. They are crucial for maintaining cellular function and communication. Defects in vesicular transport can result in diseases such as congenital disorders of glycosylation. Chromatin. Chromatin is a complex of DNA and proteins, primarily histones, found in the nucleus. It exists in two forms: euchromatin, which is transcriptionally active, and heterochromatin, which is tightly packed and less active. Chromatin structure regulates gene expression and DNA replication. During cell division, chromatin condenses into chromosomes for proper segregation. Epigenetic modifications of chromatin influence cellular function and inheritance. Nucleolus. The nucleolus is a dense region within the nucleus responsible for ribosomal RNA synthesis and ribosome assembly. It contains nucleolar organizing regions of chromosomes and is rich in RNA and proteins. The nucleolus plays a role in stress responses, cell cycle regulation, and aging. It disassembles during mitosis and reforms in daughter cells. Alterations in nucleolar function are linked to diseases such as cancer and neurodegenerative disorders. Nucleus. The nucleus is the control center of the cell, housing genetic material in the form of DNA. It is surrounded by the nuclear envelope, which contains nuclear pores for material exchange. The nucleus regulates gene expression, replication, and cell cycle progression. It contains the nucleolus, where ribosomal subunits are assembled. Mutations affecting nuclear function can lead to genetic disorders and contribute to cancer progression. Rough endoplasmic reticulum. It is a membrane-bound organelle studded with ribosomes, giving it a rough appearance. It plays a vital role in protein synthesis and modification. The RER is involved in the production of membrane-bound and secretory proteins. It also assists in proper protein folding and quality control. Malfunction in the RER can result in protein misfolding diseases such as cystic fibrosis. Ribosomes. Ribosomes are macromolecular structures responsible for protein synthesis. They consist of ribosomal RNA and proteins and can be found freely in the cytoplasm or attached to the RER. Ribosomes facilitate translation, where messenger RNA is decoded to produce polypeptides. Intermediate filament. Intermediate filaments are a component of the cytoskeleton that provides mechanical support to cells. They are composed of different proteins such as keratin, vimentin, and lamins, depending on the cell type. These filaments help maintain cell shape, anchor organelles, and provide tensile strength. They play a role in cell adhesion and resistance to mechanical stress. Mutations in intermediate filaments are associated with diseases like epidermolysis bullosa, and malfunction.
Like and subscribe the channel.